Hello and welcome to Cupcake J's Crash Course. Before I embark on my next journey, I thought I'd slow down a bit by breaking down a particularly favorite topic of mine, cell communication and cell cycle. Okay, let's start. Cells communicate with one another to exchange materials and coordinate their functions. Cell communication can be through direct contact, short distance, and/or long distance. In short distance communication, cells release local regulators into the vicinity that can be recognized by target cells. For example, neurons release neurotransmitters. In long-distance communication, the endocrine system sends out signal molecules that travel through the circulatory system to the cell's receptor proteins. An example of this would be the movement of insulin. Without the bloodstream, signal molecules wouldn't be able to travel long distances to induce a response. Now, signal transduction. Cell signaling has three primary steps: reception, transduction, and response. Reception occurs when a ligand binds to a cell's receptor proteins, causing it to change shape and initiating an intracellular signal. Then, through transduction, the signal is amplified and relayed in a signaling cascade by secondary messengers to produce a quick response. This amplification works to louden the signal made by a single ligand, making it more effective and efficient. Moving on to signal transduction. Many bacteria use quorum sensing in order to determine density of their surrounding population. High density can influence gene expression. An example of this is bioluminescence, which is the emission of light from an organism, such as the Hawaiian bobtail squid, who uses Vibrio fischeri bacteria to make this light. The nervous system in our bodies can send out these signals. There are two types of feedback systems that maintain the metabolic processes inside our bodies: negative and positive feedback loops. Negative feedback loops are responses to a stimulus that returns the system back to its target set point. For example, our response to cold environments is a shiver, which will cause our body to move and generate heat in order to warm up. This shows how important it is to homeostasis and also conditions like diabetes. When blood sugar is too low, insulin is released to break the glucose down. When blood sugar is too high, glucagon is released by the liver to release glucose. These both return the blood sugar level to normal. Positive feedback loops amplify our responses to a stimulus, setting the system further away from the initial set point. An example will include the ripening of fruit, which is due to ethylene, a hormone released during the ripening process. This is amplified when fruit is put together in a container, as the concentration of ethylene is increased, ultimately affecting the other fruits. The goal of mitosis is to create two genetically identical daughter cells. This is how unicellular organisms reproduce and multicellular organisms grow and repair cells. 2N means diploid, or having two sets of chromosomes. Interphase is composed of G1, S, and G2. The cell spends around 90% of this time in interphase. The first checkpoint is located between G1 and S, the second at the end of G2, and the third in mitosis. Mitosis is composed of four stages: prophase, metaphase, anaphase, and telophase. The details are on the screen. This ends with cytokinesis to split the cell into two separate identical daughter cells. Cyclins are proteins that bind to cyclin-dependent kinases to promote cell cycle. A functional enzyme is formed, which allows for the modification of target genes. Whenever you see kinases, think of phosphorylation and amplification of signaling in cells.